This video demonstrates the Irvine, Beatties and Bresnahan forelimb scale, or IBB, which can detect recovery of proximal and distal forelimb function. Rats are first adapted to the testing environment and observed following unilateral C6 SCI. The animals are then recorded while eating two differently shaped cereals of a consistent size. The videos are used to assess features of forelimb use, such as joint position, object support, digit movement, and grasping technique, and the presence of different behavioral features is recorded onto the IBB score sheet. The IBB shows a consistent pattern of recovery that is sensitive to injury severity. Furthermore, the IBB scale can be used to assess recovery following other types of injury that impact normal forelimb function.
Although there are several behavioral assays that are sensitive to different aspects of forelimb recovery in experimental models of cervical spinal cord injury, few techniques provide detailed information on the recovery of fine motor control and digit movement. This method may help answer key questions about recovery from spinal cord injury, like the role of particular axonal tracts in the return of proximal and distal forelimb function, for example, the rubrospinal tract or the corticospinal tract. The implications of this technique extend towards therapy of humans. The majority of human spinal cord injuries occur at the cervical level. Additionally, a key priority for patients with cervical spinal cord injury is restoration of hand and arm function. Thus, outcome measures that assess both proximal and distal forelimb functions are needed. Though this method can provide insight into forelimb recovery following spinal cord injury, it can also be applied to other types of injury that impact normal forelimb function, such as stroke or peripheral neuropathies. Individuals new to this method may